What does a bacterium getting sick have to do with genetic engineering? And why do scientists study systems that make bacteria sick? Today, let's talk about it. One of the ways that a bacterium can get sick is after infection by a virus called a bacteriophage. These bacteriophages surround the bacteria and attack it. And this attack involves injecting their DNA into the bacteria so that they can multiply themselves and spread to other bacteria. Now, to defend themselves, bacteria have a system that is kind of like an immune system. What they do is they take molecular snapshots of the DNA that has been injected by the phages. Various bacterial species use this system in which bacteria put the snapshot in their quote-unquote memory, which literally is within their DNA in a very specific place called a CRISPR array. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. Think of this in your mind like a special belt that the bacteria always carries with it that has a snapshot that is specific to the phage infection. So this is to say that the next time that specific phage appears, the bacteria is able to be prepared. Your next question might be, how does the bacteria use this information to protect itself? Now, within the genome, there are specific instructions next to the CRISPR array which are responsible for making proteins that interpret the information inside the CRISPR array. These proteins, they are called Cas proteins or CRISPR-associated proteins. Imagine this process as if the Cas proteins receive a quote-unquote message from the bacteria to help them find the specific DNA sequences that match that of the phage. And with this information, these Cas proteins take the sequence information and then they can act like molecular scissors to cut up the phage DNA, rendering it useless to the phage life cycle. In this way, the phage are unable to replicate and then they do not infect the bacterial host. The molecular details of how this process works and then the modification of this system so that now scientists could use it to edit specific areas of a DNA sequence led to the Nobel Prize in 2020. The Nobel Prize in 2020 was awarded to the two scientists who discovered a way to reprogram these molecular scissors to cut at a very specific place in the genome. We at Under the Microscope Kenya were thinking about this topic when we read a paper published in 2022 in a biology journal called Cell. In this paper, scientists reported and characterized the discovery of some CRISPR-Cas systems from phages. One can imagine that maybe the reason why phages have developed CRISPR-Cas systems might be to help them to compete with each other when many of them have infected the same bacteria. This is an interesting finding because it shows that there are more sources of CRISPR-Cas proteins than just bacteria, which means that scientists can learn even more about the diversity and breadth of these CRISPR-Cas systems, which then means better tools for therapeutic applications and gene editing advancements. This study supports the idea that discoveries about CRISPR-Cas systems are just the tip of the iceberg. Could there be more in other organisms? Me, I can't wait to find out.